Well, hello friends. Today we're going to be talking about the cell cycle and cancer. Some of the objectives: know that the cell cycle is a repeating sequence of cellular growth and division during the life of an organism. Understand that cells divide to reproduce, in the case of unicellular organisms, to grow and to replace worn out or damaged cells. Describe the stages of the cell cycle. Know that the cell cycle in eukaryotes is controlled by proteins at 3 main checkpoints. Recognize that if any of the genes necessary to make the proteins that regulate cell growth and division are mutated, the protein may not function, and the regulation of cell growth and division may be hindered. In the table of contents, we're going to be talking about an introduction, reasons for cell division, chromosomes, karyotypes, cell cycle summaries, specifically interphase, mitosis and the phases therein, cytokinesis, controls of the cell cycle, cyclins, CDKs, checkpoints, cancer, and finally, a summary. An introduction. All cells are derived from pre-existing cells. New cells are produced for repair to replace damaged or old cells, and it differs a little bit in prokaryotes. They do a process called binary fission. Eukaryotes, however, perform the cell cycle. This would include protists, fungi, plants, and animals. Now, there are a couple of different reasons for cell division, one of which, and a major one, is called cell growth, for the repair and replacement of damaged cell parts. Also, reproduction. Some species reproduce through cell division. Different types, we as animals, reproduce through sexual cell division, also known as meiosis. Why do cells need to replicate their genetic material or chromosomes before going through mitosis? Well, the instructions for making cell parts are encoded in the DNA, and each new cell must get a complete set of the DNA molecules. So how do we replicate? What is DNA replication? Well, DNA must be copied or replicated before cell division. We must make a brand new set of the old copy. So we must replicate DNA or make a brand new set of DNA for the new cell. Each new cell will then have an identical copy of that DNA. Now, does DNA look like the way that we've portrayed it? And the answer is yes. However, it is coiled up into what are called chromosomes. Now, in animals, and specifically human beings, we have 46 chromosomes, 23 from the maternal, and then 23 from the paternal side of that organism. Now, chromosomes, let's talk about the structure for a moment. DNA is tightly coiled around proteins called histones, further condensing that forms that chromosomes, as you can tell in the picture right. Now, when replication occurs, these chromosomes replicate. They go from one chromatid to two identical chromatids called sisters. They are attached in the middle of the two chromatids by what's called a centromere. When the chromatids are drawn apart toward opposite poles during anaphase, the chromatids are then separated or pulled apart. Now, what is a karyotype? A karyotype is a picture of the chromosomes from a human arranged in pairs by size. We use karyotypes when we do forms of genetic testing to see if there are any chromosomal disorders. Now, the first 22 pair are called autosomes. So if you look at all of these pairs, the first 22 pair are indeed autosomes. The last pair, this pair right here, is called sex chromosomes. They're either XX for female or XY for male. And if you look at this set, you can see that you have XY. Now, the cell cycle summary. Let's talk about how it starts. The first phase is called interphase. Now, it is part of the cell cycle, but not a part of mitosis, which we'll get into in a minute. There are specific parts of interphase that you need to know. So 5 phases of the cell cycle go like this. Interphase incorporates or encompasses G1, Gap1, or primary growth phase. There are 3 names for this one phase. First growth phase after cell division is cells mature or they make more cytoplasm or organelles because remember they're preparing to double up in size, eventually to divide. Cells carry on as normal metabolic activities until we enter S phase. Now S phase, also known as synthesis phase, is where DNA is copied or replicated. Probably the most important phase because when you're copying DNA, this is when copying errors or mutations can occur. Now in G1, G2, or secondary growth phase occurs after DNA has been copied and all cell structures needed for division are made. The centrioles, both organelles and proteins, are synthesized, certain enzymes to continue cell division. Then we head into mitosis. So mitosis, the M phase, proceeds directly after interphase. Now, what is mitosis in general? Mitosis, by definition, is called the division of the nucleus. It is also called karyokinesis. Remember, karyo is nucleus. Eukaryotic organisms has that karya in it. That's the nucleus. Also occurs in eukaryotes, has 4 stages, and doesn't occur in some cells, such as brain cells or neurological cells. There are 4 stages indeed. The first stage, and there are several different stages within those, is called prophase. Now in early prophase, chromatin, thread-like DNA in the nucleus, condenses to form visible chromosomes. A mitotic spindle also forms. Now what is this? These are aster rays and spindle fibers that project or protrude out of the centriole. And then in late prophase, nuclear membrane is broken down. You'll see the nucleus begin to disintegrate, and the nucleolus are also broken down. Chromosomes continue condensing and are clearly visible in late prophase. Spindle fibers called kinetochores attach to the centromere. These spindle fibers work their way down and start trying to attach to the centromere of each chromosome. After the mitotic spindle finishes forming, the mitotic spindle forms from these microtubules in plants and centrioles in animal cells. Some polar fibers, some portions of the spindle fiber extends from one pole to the cell to the opposite pole. Kinetochore fibers extend from the pole of the centromere to the chromosome to which they attach, and then these aster rays are short fibers radiating from the centriole. It looks like this, and you can see early prophase, not a lot of chromosomal structure, but by the time we get to late prophase, you will see these are the aster rays on the outside. You will see the kinetochore fibers starting to form up at the centriole or at the centromere of the chromosome, and the centrioles at the opposite pole start to line up these sister chromatids in the middle of the membrane, or the middle of the cell. Now we have what's called metaphase. Now I always tell mine, meta and middle, start to connect those terms in your head. I often say alliteration is your best friend when we're talking about biology. So meta and middle. Metaphase, these chromosomes that are attached to the kinetochore move to the center of the cell. So you can see them starting to align at what we call the equator or the equatorial plate, or the metaphase equatorial plate. Chromosomes, they're lined up there to show this kind of structure. After metaphase, you have what's called anaphase. I tell mine that anaphase means apart. So you can see these sister chromatids starting to be pulled apart by the shortening of these spindle fibers. So sister chromatids are pulled apart to opposite ends of the poles by shortening the kinetochore fibers. Polar microtubules extend, preparing for cytokinesis. And then finally, telophase. All of a sudden you start to see 2 distinct structures of cells. You also see the cytoplasm here in the middle starting to pinch. The sister chromatids have moved into the opposite poles. The spindle fibers start to disassemble or disappear. The nuclear envelope, this gray structure here in the picture, starts to reform around those chromatids. The nucleolus reappears on the inside, and then what is the next phase called cytokinesis begins. Now, the chromosomes appear once again not as shortened condensed chromosomes, but more like that thread-like chromatin. And then finally we have what's called cytokinesis. Cytokinesis means division of the cytoplasm. It's where the division of the cell into 2 identical halves called daughter cells. Now in plant cells, the cell plate forms at the equator, so this would be a plant cell, and you'll see this division line starting to form. We call that the cell plate, which eventually becomes the cell wall of plants. Animal cells don't have a cell wall, but they have what is called a cleavage furrow. This cleavage furrow helps split the cell. So make sure you understand the difference between how plant cells and animal cells undergo cytokinesis. And then you have two complete daughter cells at the end. Once this connection is broken, they have the same number of chromosomes, so they are identical to one another, but a little smaller in size to the parent. So they must grow to become mature cells in G1 or G1 of interphase. Now, there's some controls of the cell cycle. This doesn't happen by happenstance. The importance of cell cycle regulation to properly control how the cell cycle is very important in maintaining a balanced and healthy growth of the cell. When the cell cycle goes awry, it can lead to diseases such as cancer and or developmental disorders. So the key players are what are called proteins called cyclins. You can see a couple here, G1 cyclin, G1S cyclin, S cyclin, and the M cyclin. Also CDKs, which we'll talk about here in a moment, and the cell cycle checkpoints are the main components that regulate the cell cycle. Now, what are cyclins? There are a couple different types. One is called the cyclin-dependent protein. Now, these are special proteins that regulate the activity of other proteins called CDKs, which we'll talk about here in a minute. They are named cyclins because their levels in the cell go up and down, thus the term cycle, in a repeating pattern during the cell cycle. We also have what are called regulatory proteins. Now, these cyclins control the process of the cell cycle by binding to and activating specific CDKs, and they work like helpers for CDKs, enabling them to do their job of adding phosphate molecules to specific proteins involved in the cell cycle. So what are the CDKs? They are cyclin-dependent kinases. They attach themselves to the cyclins attach themselves to CDKs, making them active and ready for work. So the cyclin-CDK combinations adding phosphate molecules to specific proteins and help the cell move through different phases of the cell cycle. Now, there are some transitions that happen during the cell cycle: prophase, metaphase, anaphase, telophase, cytokinesis. These cyclins make sure that each phase of the cell cycle is completed before the cell moves to the next phase. Different cyclins are active at different times during the cell cycle and ensures that each phase happens at the right time. So the first one we're going to talk about is G1 cyclin. So obviously it happens during primary growth, or G1 or GAP1. They help the cells move through the first phase of G1 to the DNA synthesis phase by activating certain CDKs. One of those CDKs or cyclins that we're going to talk about is the S-phase cyclins, or cyclin E. S-phase cyclins help start the process of copying the DNA by activating other specific CDKs. We're not going to get into the detailed specifics of the CDKs and their molecular makeups at this point. Now, the mitotic cyclins, or cyclin B, help transition from the growth phase to the division phase, or mitosis: prophase, metaphase, anaphase, telophase. So in this little section, we've talked about G1 cyclin, or cyclin D. We've talked about cyclin E, or S-phase cyclin, and we've talked about cyclin B, or the mitotic cyclin. So we've moved through the cell cycle from interphase to cytokinesis. Now, the cyclin-dependent kinases, they are specific enzymes that add a phosphate molecule to proteins. You can see how they're working their way through here. And you can see the G1 or S-phase cyclin that hooks up, and then you start S-phase, you degrade that CDK, or that cyclin. The CDK remains, it hooks up to the next S-phase cyclin, then it breaks it down, hooks up to the G2 cyclin, breaks it down, and so we process or progress through interphase in that way. Now, activation, they're binding to those cyclins specifically to form these active pairs, so like the enzyme substrate processes that we were talking about back when we were dealing with macromolecules. Here are some specific CDKs. CDK 4 and 6 occur for G1. 4 and 6 with cyclin D occurs for G1. Cyclin CDK2 is in S phase. along with cyclin E, and then CDK1 in G2 along with mitosis. So once again, these CDKs are going to be kinase. That kinase should indicate to you that they are enzymes, and they hook up with the cyclin itself to make that giant molecule to help precede cells through interphase. Now, there are checkpoints that occur here. There are specific control points. There's G1, there's GS, there's G2. There's also a mitotic checkpoint that we're going to talk about. Then we're going to talk about G0. So, definition and importance. Specific control points. These checkpoints are like checkpoints in the cell cycle where it stops and checks to make sure everything is going well before moving forward. They are crucial to ensure that the cell divides correctly and doesn't make mistakes. So, preventing cell cycle progression under unfavorable conditions, these checkpoints should stop the cell cycle if they detect damaged DNA, for instance, or unfavorable conditions for growth, temperature, pH, all of those things. It'll pause to fix those problems before moving on. Now, we need to talk about what happens if they don't, and so that's coming. So there's the first checkpoint, G1 or S checkpoint. It's a role in assembling DNA integrity or assessing that DNA integrity in growth conditions. It checks to see if the DNA is intact, has been copied correctly, the cell has the necessary conditions for growth, the cell is actually doubled in size, has enough organelles, cytoplasm, all of that good stuff. There is an activation and inhibitor or inhibition of cyclin CDK complexes, so it will stop right here if those conditions are not met. G2 and M checkpoint, after S phase where synthesis or replication has occurred, that G2 checkpoint makes sure the DNA replication is finished, detects any kind of DNA damage or miscopying, inhibits or activates that cyclin CDK complex, so it'll either move forward or stop. Then you have a metaphase spindle assembly checkpoint, making sure that those spindle fibers are attached correctly. If they're not, then you'll never proceed into anaphase, pulling those sister chromatids apart. Then the cell does not have the requisite number of chromosomes that it's going to need to be a functional cell. So it can delay anaphase onset until all chromosomes are properly aligned. DNA damage checkpoint, role in detecting DNA damage and activating repair mechanisms. DNA polymerase, a polymerase that we'll talk about in the next unit when we talk about DNA replication. That DNA polymerase goes back and checks for errors and then acts as an editor. So if you're familiar with an editor for a book, they go back and check for mistakes or grammatical errors or those kind of things. This is what DNA polymerase does. And then it'll activate either cell cycle arrest or DNA repair pathways. Cell cycle arrest is saying, hey, you're not going through. You have enough damage where we have a problem. Now, what happens if none of that takes place, which is a thing? We have what is called uncontrolled mitosis and or cancer. If mitosis is not controlled, unlimited cell division occurs, causing cancerous tumors. A tumor can be benign or it can be cancerous, and we'll talk about that here in a minute, and metastasis and those good things. So at checkpoint G0, the cell has a decision to make. Keep replicating or go perform its function, or the third is what's called apoptosis or cell death. If the cell is beyond normal replication cycle and has enough mutations and it keeps dividing, however, that is cancer. Now, what are oncogenes? They're specific proteins that increase the chance that the normal cell develops into a tumor cell. Now, what is cancer specifically? It's essentially a disease in mitosis. The normal checkpoints regulating mitosis are ignored or overridden by a cancer cell. The cancer cell then begins with a single cell that is transformed or converted from a normal cell to a cancer cell. Often this is because of a change in the function or a mutation that occurs in one or several genes that normally function to control cell growth. So some examples of one of these genes is the p53 genes. It's the guardian of the genome, usually functions to properly control the cell cycle. However, it is mutated in over 50% of human cancers. So once you mutate the p53 gene, it is possible that it turns off the controls necessary for controlled cell division, turning it into a cancer situation. The BRCA or BRCA gene is the breast cancer gene, normally functions to suppress tumor formation. But if the gene contains mutations, it causes it not to work, therefore tumor formation can begin. One of these start behaving abnormally. Cancer cells start to proliferate, meaning grow, spread wildly by repeated cell division. So right here in this CT scan, you can see on the right this tumor that is growing, and you can see some linkage into the surrounding tissue that is growing in the brain. Now, the cancer cells proliferate or grow to form a mass of cancer cells called a tumor. It can be benign, okay, meaning it stays at its original site, or it can be what's called malignant. Some tumor cells send out signals to tell the body to produce a new blood vessel at the tumor site. And once these tumors have blood flow, then tumor cells can spread throughout the body. That spreading is called metastasis. Cancer cells are frequently known as immortal, whereas normal cells divide about 50 times and die. Cancer cells can divide indefinitely if supplied with nutrients, thus the blood vessel that it tries to get to grow next to the tumor or to infuse the tumor with new blood. Cancer cells often have unusual numbers of chromosomes or mutations in the chromosome. Aging produces these situations. Exposures to toxins, mutagens, all cause mutations in genes or cancer, but normal errors in DNA replication can lead to the actual transformation or mutation of a crucial gene causing cancer. Cancer cells may also have an abnormal cell surface. Instead of being smooth and it moving past, it can be sticky, so it connects to nearby cells, and they round up and they break attachments to neighboring cells, following or allowing for metastasis. So cancer cells ignore the usually density-dependent inhibition of cell growth in a cell culture or body tissues. Multiplying after contact with other cells are made. Usually cancer cells, when they come into contact with other cells, they tend to stay localized. However, if they have these sticky surfaces, they multiply upon contact regardless of how dense, how many cells are packed into a finite spot. How do we stop cancer? Well, there are a couple different ways. Chemotherapy drugs stop DNA synthesis and replication. Here are a few that we use for chemotherapy. Now remember that chemotherapy, these types of drugs also kill healthy cells as well. So not only does it take care of your cancer cells, but it also affects healthy cells. That's why the side effects of these types of drugs are pretty hard on the human body during that time. So there's also radiation therapies, and there's also surgery if possible. There are some tumors you can't get to. There are some tumors that are in areas that if you perform surgery, it can damage the organism in general. Same thing with radiation. So, cell cycle, a summary. Why does it happen? Well, growth and repair. When does it happen? When does mitosis, the cell cycle that is asexual reproduction, it occurs from fertilization until death. So when the sperm and egg come together, make the zygote, from the zygote process on, that zygote from 1 cell to 2 to 4 to 8, and it grows, it doubles every time. That is mitosis, and it occurs until death. Where does it happen? So you'll see the term somatic. It means body cells, skin, muscle, bone, tissues. It doesn't happen in the brain. And there are some spinal cord injuries that we are finding with new research that we used to think that spinal cord cells, the actual cells themselves, will not regenerate. Now with some specific therapies, they're doing some breakthroughs currently on spinal cord research. So that might be coming soon. So what are the outcomes? 2 genetically identical daughter cells. Something to keep in mind here. So ladies and gentlemen, we've talked about the cell cycle. We talked about the phases that are contained within the cell cycle proper, also known as interphase, which makes up G1, S, and G2. We talked about mitosis, prophase, metaphase, anaphase, telophase, what happens within those cells during those specific phases. And finally, cytokinesis. We talked about the controls of the cells, the cyclin-specific proteins that control different phases of the cell cycle, and CDKs, the enzymes that hook up to those controls, those cyclins, and make the CDK cyclin complex and maneuver those cells through those specific phases without issue. We talked about what happens if there is indeed an issue. At those checkpoints, G1S, G2, metaphase, those checkpoints, if it doesn't find or identify specific problems in DNA replication, or the cell just grew in conditions where it wasn't optimal, and it allows those cells to progress through, it can be an uncontrolled mitosis situation, which we refer to as cancer. We talked about oncogenes, how to treat and effectively try to treat cancer. And then finally, we wrapped it up with a summary of the cell cycle. Why, where, when, and how, and what are the products of the cell cycle. Ladies and gentlemen, if you have any questions, comments, or concerns, make sure you address it with your instructor. Have a wonderful day.